Hello, welcome everybody to this podcast series. We have three very interesting speakers here today with me. My name is Amaia Zurutuza. I'm the scientific director of Graphenea, and I will be moderating this podcast. So these three speakers, they are all involved in three very interesting projects within the Graphene Flagship. And we want to discuss a bit and learn more about their individual projects. And the podcast is about graphene biosensors in healthcare. So let's start. Okay, here we have Spyros, that he's involved in the Graphergia project.
Thank you for having me. Thank you to Graphene Flagship for hosting this event. It's very nice to be here and having this conversation with all of you.
Great, welcome. And then we have Alexey. He's the coordinator of the Munaset project. Welcome, Alexey.
Hi, Amaia. Thanks for having me here. I'm Alexey Tarasov, coordinator of Munaset, and I'm physicist by training, and I'm also professor of biomedical engineering at the Kaiserslautern University of Applied Sciences. And I'm glad to be here.
Great. And last but not least, we have Vincent from Graphill, and he's working in the project 2D Biopad. Welcome, Vincent.
Thank you, Amaia, for having me. I'm Vincent, so I'm the co-founder of Graphill. Graphill is a part of the 2D Biopad project, and we are doing a graphene polymer biosensor for this project.
Okay, great. So let's kick off the discussion. So what we want to know is to learn more about each of these projects. So let's start, for example, with Graphergia. So what is the motivation behind the project?
The core motivation is to create autonomous and self-powered sensing mechanisms, which have the ability to be battery-less, no need for batteries, no need for charging, through textiles. So the clothes we wear, we want to energize them and functionalize them, for them to function as an energy supplier and as a sensor. That's basically the aim that Graphergia promises to deliver.
Okay, great. And in the Munaset project, Alexey, what is the motivation behind the objectives, the aims?
So the main motivation in this project is depression. So depression is a big burden for our healthcare. So it's a severe depression which affects more than 20 million people in the European Union. And it's also very costly brain disease. So it's a great burden for patients and for the entire health system. And one problem is that currently there are no blood-based tests to actually help to guide the therapy. So there are therapies available, but they are not really personalized. And it's difficult to find the right therapy for patients. So it's basically based on trial and error approach, which takes months, which is also difficult for the patients and also quite expensive for the entire society. So therefore, our goal here is to develop new blood-based biomarker tools to actually help doctors to find this therapy faster and towards personalized medicine.
Okay, great. And last but not least, Vincent, can you mention a bit about Biopad, the motivation behind the project, the objectives?
Yes, so the 2D Biopad is tackling another neurological disease, which is affecting also a lot of Europeans, which is the Alzheimer disease. Again, for Alzheimer disease, we don't have blood screening tools. There are appearing the first clinical lab tools, but what we want to deliver with 2D Biopad is a screening point-of-care tool that can be deployed in nursing home. And so we can actually screen at early stage Alzheimer's disease, because we know that if we take at early stage this disease, we can stop or delay the appearance of the most serious problem.
Okay, so listeners, you are listening to three new technologies that are really, let's say groundbreaking, because they are really fitting a market need. There is nothing available currently in the market. So now we want to find out a bit how you are going to achieve these ambitious aims, let's say. And we can start with Vincent now.
So it's as you can imagine, it's a very challenging problem, because we actually combine two challenging technologies. The first one is to deliver a new type of sensor, which has never been on the market yet, even for simple disease, which is the biosensing based on graphene sensors. I will maybe go into much more in the details, but there are two types of sub-technology based on graphene, which is developed in the project with two different research groups. But the second challenge on top of that, the first one, is actually to detect a set of biomarkers which are present in blood at very tiny amounts. As you know, there is a significant barrier between the brain and the blood. And so the biomarkers which are significantly present in the brain, which are the hallmark of Alzheimer's disease, they are actually present in the blood, but with 100 times smaller amount due to these barriers. And so we have to detect at sub-femtomolar the amount of these biomarkers. And on top of that, it's not only one, it's 5, because one is not enough. And so it's the set of the 5 value, quantitative value, which give a picture of if you are subject to early stage of Alzheimer's disease. So it's very challenging project.
But maybe the question is, how are you going to do it? Like, what are you going to use to achieve?
So the way to do that is that we combine probes which are already known, such as antibodies and aptamers, mostly aptamers, which we developed during the project with an aptamer developer, which is part of the consortium. And these aptamers are small DNA strands, which actually coil and create a hook that are able to attract and bind on the surface of graphene. And this binding is giving a signal we detect, even at tiny amounts. And this binding creates a distortion, such as the change of resistance in the graphene layers.
Okay, so the keyword here is graphene for the moment. Okay, what about Munaset, Alexey? How are you going to do it?
So we want to measure biomarkers similar to the previous project. So we also are targeting a panel test, so several biomarkers, because one biomarker is usually not enough, so it's not specific enough. And in our case, these are different biomarkers. So these are enzymes called proteases. So they are everywhere in our body, and they are also involved in this depression. And actually, the levels of different proteases can be increased or decreased during the therapy. So we can, for example, later on compare these patients with and without treatment and see what the results are and how these biomarkers behave. Now, about our technology, so it consists of several parts. So first of all, our sensor needs to be very sensitive. This is why we use graphene field effect transistors. So these are highly sensitive devices that can detect charged species. The sensors need to be also selective. So to achieve selectivity to different biomarkers, we need custom-made peptides that we designed during this project. And these peptides, they can interact with proteases. And as a matter of fact, we are measuring the activity of these proteases, which means that they can actually cut these peptides that are bound on the surface of graphene. And therefore, we see a signal change if there is a protease in the sample. And third point is we need to be also, our sensors need to be stable and reproducible. And this is why we also want to integrate them on a readout chip. So we have a multi-analyte detection system. So there are many different graphene chips that are very reproducible and stable. So these are the 3 components we need to get this going.
Okay, and for the wearable self-powered sensors, how are you gonna achieve this type of sensors?
The aim is to have an autonomous device. So the first thing you have to look at is how to implement it on an everyday basis, so as not to disrupt the patient's life and normal way of living. So you have to add a non-disruptive apparatus. So it has to be really thin, really lightweight, and all that. So what we've designed is a prototype, a way to functionalize textiles. So you place graphene on top of everyday clothing, which in turn can generate energy, and then via sensors based on graphene triboelectrically, with triboelectric nanogenerator effect, generate enough charge to power the sensors themselves, so as to have a monitoring of heartbeat, a monitoring of the gait of a person, the way they walk, which can then be preemptive as a not as a therapy, but as a indicator that somebody might have a problem in the future. So the way we functionalize the textiles is based on graphene, as I said, and we want to create a non-disruptive apparatus which can be applied on everyday clothing as we know it.
Okay, perfect. And maybe now I think it's important also for the audience to know a bit about the impact, right? I mean, what is the expected impact of each of these three different, let's say, technologies, right? So maybe we can start with the
Right,
Spyros.
sure. Yeah, as we know, to have a clinical diagnosis, you need to go to the doctor and have a checkup every now and then. So what the application of Graphergia aims to do is to bypass that aspect of the whole deal. So you have a continuous device that continuously can do the diagnosis preemptively for cases such as cerebral palsy, maybe spinal problems or walking problems, and even Parkinson's disease and all that. The impact is multifaceted because it affects people of all ages, mainly the elderly, of course, who have a big problem with cables, batteries, and changing and all that. So the autonomous factor is at play there. But it will also help with people such as athletes and whatsoever.
Okay, very interesting. What about in the Munaset project?
So the goal of the project is to demonstrate the sensing principle for several proteases. This is what we want to do. So we started at technology readiness level 3, and we are going to move it towards 5. What does it mean? This means that our sensor will work in a lab environment. So this is our project goal. And beyond that, of course, we envision that these devices will be developed further. In the end, of course, we have our vision is to have a small device, portable device that can be used at the point of care to help doctors monitor the therapy of depression and other psychiatric diseases. Beyond that, I think in general we have a very flexible platform that can be also adapted to other biomarkers, especially if we use the peptides I mentioned. So we can also synthesize other peptides. So there is many options. So we can also contribute to diagnostics, for example, of other diseases, including cancer. And beyond that, it's also interesting for us to study, for example, protease inhibitors. So it's a related topic, and this can be interesting for drug screening applications in the future. So these are the impacts that we have in mind.
Okay, very good. And for the 2D bio part?
Yes, so the impact is indeed to have a better screening of the population. Actually, there are many types of dementia. And actually, Alzheimer's disease is rather underdiagnosed in Europe. And as I said, if we can find, even before the first symptoms, the precursor of this disease, we can really act in a more decisive way and delay the most problematic symptoms. So that's, I think, a very important impact. Nevertheless, like in Munaset, we are in TRL 4 at the moment. At the end of the project, it's going to be 5. We have 2 clinical trials which are scheduled, and the first one is going to happen in 6 months from now. And at the end, it's more. It's not going to be a diagnostic device. I think we're not intended to do that. It's more going to be a screening device, which has a bit different ethical implications, because we don't want to say after this test that you have the disease. It's more like having a pattern which need to be confirmed with more advanced diagnostic device, such as brain scan, for example. And so that's an important point, because, of course, it's a very serious disease and the ethics has to be considered.
Okay, so if I understood correctly, so the idea is also to decrease the regulation burden, right?
Correct.
So more as a monitoring more than a medical device, right?
It's a screening device, even though it's for to be marketed in EU, you need to pass the stringent medical device IVD, in vitro diagnostic.
Okay.
Nevertheless.
Okay, and maybe now talking a bit more into the challenges. So, I mean, these projects have been going on for a few years, not many. We still have 1 year to go, right? And so what are the main challenges you have faced during these past years? And what would be the challenges to see this type of, let's say, sensors in patients in the real world? Who wants to jump in?
I could, yeah,
Okay,
sure, for sure.
come on.
The thing is that, as to reiterate what I said before, our function is to energize clothes, basically. But the thing is that every piece of clothing is different as its substrate. Some are polyester, some are polyethylene, some are whatever you may have. So you have to differentiate the parameters of graphene deposition on each piece of clothing, which will then at a later stage hit the market. So that's a big challenge to find the actual pairing of parameters needed for each piece of clothing, so that it's not constricted, like the people who will use it tomorrow, the day after tomorrow, metaphorically speaking. They don't have to use one type of clothing. They can implement it in whatever piece of clothing they wear. They don't have to care about that as well as their health problems. That's a big challenge to create pairings of manufacturing processes and optimize these couplings.
Yeah, and also another thing I think of is washability, right? I mean,
Exactly.
this must be a hot topic for textiles because we wash them very often, and yeah.
Of course, you have to have a way to encapsulate the process you've created on top of the textile and make it durable and make it long-lasting. So to negate problems such as the one you presented right now. So it is challenging. It is multifaceted. It's like the colleague said before. It's also aiming at TRL 5 and a pilot level, but we're doing the best we can, of course, as always, and it's looking good.
Yeah, regarding this Alzheimer disease screening device, the main challenges is to reach the sensitivity level which are needed to detect it, because we need to be quantitative. It's not 0 or 1 like in COVID when you don't have the protein of the virus or you have it. In the case of this biomarker between a healthy patient and the disease, it's just a factor 2 at the femtogram level. So it's really, and you need to be quantitative with a cutoff well defined. And that's not only on 1, but on the 5. And that's very challenging because if we cannot reach one of these limits, the system will not work correctly. And we cannot afford that for such a very serious disease. So that's, I would say, the first biggest challenge we face now.
Okay. Alexey?
In our case, selectivity was one of the challenges in the initial phase of the project. These biomarkers that I mentioned, these proteases, I mean, they are quite similar. Yeah, even though these are different molecules, they are still quite similar in terms of structure. So it is not easy to find peptides or capture molecules that can interact selectively. So this took us a while, several optimization steps to develop, to synthesize these peptides, to characterize them carefully. And we already published several scientific papers about this topic, about these peptides and also the initial assay. So I think we have solved these challenges now. So we have several peptides now that are optimized towards these biomarkers. And one of the next challenges will be to measure actually clinical samples. So we have already collected clinical samples from patients with depression that also had some treatment, and also control samples that don't have depression or didn't have treatment. So this is also one of the next steps to characterize carefully these clinical samples with reference methods, with gold standard methods, and then also validate our biosensors using these clinical samples. So this is one of the next challenges we face in this project.
Okay, and maybe now to compare a bit what is the situation, let's say, in Europe or in the rest of the world. I mean, what do you think about your technology? How is it positioned? Is Europe well positioned, or are we losing to race versus Asia or the United States or other parts of the world? Vincent, what do you think?
I think for graphene, at least, there is a task force in Europe which is well organized, thanks to all the 12 years of work of this Graphene Flagship. I would say that's put really Europe in a very nice position. Nevertheless, I would say I'm traveling quite often in Asia, in South Korea or in China, and I'm very impressed by the progress they're making in this subject as well. And in China, we don't realize how much they also invested in graphene. And they do have both academic excellence now and industrial knowledge and industrial transfer, which is also very impressive. So I would say, yes, in Europe, we have a very good position, especially on this biomedical application, I would say. But in China, it's going very well also. And in Asia, we're going to see a large development in the coming years.
Alexey, what do you think?
I think Europe is well positioned in this field. So our sensors, I mean, they really need some interdisciplinary collaboration. Yeah. So the components of our sensor, they are all made in Europe. Yeah. So we have partners across Europe who contribute to this project. Yeah. So we have graphene, the raw material fabricated here. So we have the peptides fabricated here. We have the integration done in Europe as well. So all the components, so the entire value chain is made in Europe, which is, I think, great, and it's possible because of this big Graphene Flagship funding that is available here. Nevertheless, I mean, of course, I have to agree with Vincent. I mean, there is competition, and we have to be careful. Yeah, so we need to move fast to get this towards higher TRL levels and to the market.
And Spyros, what do you think about the smart textiles or monitoring health?
Our take is aligned with what Vincent referenced, the fact that it's been a push and pull relationship between Asia and Europe, and who gets the head start and who is not. I think during the past few years, the past 1 and 2 years, it's gotten to a point where Europe is a very close second. The innovation happening over there is an aspiring one, of course. The thing is that in Europe there are more forefronts of development. The issue with the consortia we have, is that you have many different viewpoints in the same project, which can promote development and projects in general inside the Graphene Flagship initiative, which I think will help us in future to get the head start once more.
Yeah, I think people listening, I think you can see that collaboration seems very critical in Europe. So we need to be together on this in order to make it happen, right? And improve patients' life, European patients' life. So I think we can maybe close here the discussion. I don't know if you have any last points. Take-home messages? Vincent?
I think, the consortium is key, and not only on the technology, but also on the clinical levels. In the case of 2D Biopad, we have 3 clinical level at 3 places across Europe, Finland, Germany, and Greece. And of course, the input and the feedback from the clinician is key for this kind of healthcare project. So it's also thanks to this networking of the clinics, and which is also very important.
Okay, so thank you for listening, and watch this space.